Okay, we are starting with the discussion of this experiment. This is activity 11.3, investigating the production of carbon dioxide by anaerobic respiration. Now, before we go through this experiment, I want you to give me the meaning of anaerobic respiration. What is the meaning of anaerobic respiration?
Anaerobic respiration is a process which happens in the absence of oxygen. It usually is
See,
different in plants and
basically, respiration is a process in which glucose or any food substrate will get used up and there is going to be release of energy. When you say anaerobic respiration, it means there is absence of atmospheric oxygen. Just don't say absence of oxygen. Oxygen will be there, but atmospheric oxygen is not going to be there. There may be different organisms under different conditions that will be carrying out anaerobic respiration, and different products also will be formed during the course of anaerobic respiration. Okay, for example, if certain here this example that we are considering, the experiment that we are having yeast in these experiments. Okay, now if it is subjected to certain conditions depending upon the substrate food that is present, we may get different products. Carbon dioxide is one of the products that we are going to get. The other product can be alcohol, ethyl alcohol or ethanol. Okay, but at the moment we are not studying that. We are just studying over here what is formed during the process of respiration. It is the gas carbon dioxide. Okay, so we'll just go through this. What are the skills required? Using techniques, apparatus and materials. What is planning, observing, measuring and recording, interpreting and evaluating observations and data. So we'll just go through these experiment that is given to us. Okay, the first statement is boil some water to drive off any dissolved air. Okay, because here we are studying the process of anaerobic respiration, there should be absence of air or oxygen. So first, some dissolved air is present, oxygen is present in water. So we will boil off that and we are going to remove that dissolved oxygen. Then dissolve a small amount of sugar in the boiled water and allow it to cool. So the sugar which is there, it is going to be the food substrate which is going to undergo anaerobic respiration. When it is cool, add yeast and stir with a glass rod. This yeast which is there, it is going to give us the enzymes, the biological catalyst which is going to carry out this process. Set up the apparatus as in the diagram. Add liquid paraffin by trickling it gently down the side of the tube using a pipette. Okay, we are going to have a look at these apparatus. We are also going to see the reason that of course will be there in questions part, why we are adding this liquid paraffin. Okay, then the next part is set up the identical piece of apparatus but used boiled yeast instead of living yeast. So similar apparatus is set up, but there we are using boiled yeast. Leave your apparatus in a warm place, so sufficient warmth will be required so that appropriate temperature is available to us for the experiment. Observe what happens to lime water after half an hour. Obviously, you know lime water test, it is test of carbon dioxide. If carbon dioxide is evolved, in that case lime water is going to turn milky or it is going to turn cloudy. Now we are going to look into this apparatus. Okay, just see what this apparatus is. We are having here a cocked test tube is there. Okay, what we have taken over here, yeast is taken, boiled water is there to drive off oxygen or air, and cooled sugar solution. This is one set of apparatus. In the second set of apparatus, we are taking boiled yeast and other things are remaining same. And we are leaving this in the warm place. Whatever these gases will be formed, that is made to pass through the delivery tube through lime water. Okay. And of course, this tube is there so that whatever the excess gas which is there, it can escape out into the atmosphere. Okay, now we are going to answer these questions. So these are the questions that we are answering. Why it is important to boil the water? Can you state the reason for this?
Yeah, it's important to boil the water so you dry off the dissolved air around.
Correct. Okay, you remove the dissolved air because we are studying anaerobic respiration, so presence of air is not required over here. The next question, why must the sugar solution be cooled before adding the yeast?
Oh.
Why should we use
It should be cool because when the yeast reacts, if it's hot, it won't react well.
No, it won't react. We don't say exactly that. Okay. Now, this last time in the chapter of enzymes, we had discussed. Okay, yeast is going to be active only up till a certain temperature. Beyond that, what is going to happen to this yeast?
Denatured and like, yeah.
That's correct. It will get denatured. Therefore, this part is necessary. It has to be cooled. Otherwise, the yeast will get denatured. We are answering the next question.
Yeah.
What is the liquid paraffin used for? This is the top surface of liquid paraffin that is formed. What is the purpose of using this? Why can't we use these experiments without that? Why can't we carry out without that? What is the purpose of this liquid paraffin?
Liquid paraffin, so whatever, like nothing will escape, the CO2 or the oxygen won't escape and it keeps the substance.
No, you see, whatever the CO2 that is formed, that is going to go and it is going to get bubbled through this. CO2 is going from here. It is passing in this direction. It is going like this. And it is passing through lime water.
Yeah.
Okay. See, the purpose of adding this liquid paraffin is here. You can see over here in this part also some air is present.
Yeah.
It's not vacuum. So that air should not come in contact with yeast and sugar.
So external air shouldn't react.
Yes, this liquid paraffin which is there, it is forming a barrier so that whatever air is present over here, that also does not reach down.
Yeah.
That is the purpose of keeping a layer of liquid paraffin on the top surface of this liquid in which we are carrying out this experiment. Okay, so the answer that we are going to write for this is liquid paraffin forms the barrier for air or oxygen which is above the solution in which experiment is carried out. It forms a barrier for oxygen or air. Okay. Then next part is what happened to lime water or hydrogen carbonate indicator solution in each of your piece of apparatus, what does this show? Okay, obviously we know, we have discussed this just now before only. Lime water is going to turn cloudy. Okay. So obviously this carbon dioxide that is formed, it will get collected over here, that is made to pass through lime water. So this lime water solution is going to turn cloudy. This experiment shows that carbon dioxide is evolved in the process of anaerobic respiration. One more part that we are discussing over here is this hydrogen carbonate. The solution that we are going to take over here is hydrogen carbonate. Okay, basically it is going to be calcium hydrogen You were asking something?
No.
Okay.
5 and 6 we don't have to submit, and we have to do 11.2 also.
Yeah, we will do that. Okay,
Yeah.
what new substance would be produced, would you expect to find in sugar solution containing living yeast at the end of the experiment? What new substance would, so it can be alcohol. What is the other product that is formed? This can be alcohol. Describe a method you could use to compare the rate of carbon dioxide production by yeast using different kinds of sugar. Remember to describe all the variables you will change, those you will control, and how you will collect, record, and analysis. Obviously, the rate of carbon dioxide formed we are supposed to get. So what is the kind of apparatus that we can use?
You can use the same like the test tube and the paraffin and everything, and you can change the types of sugar.
That's
Like
correct. How are
so
we going to measure the rate of amount of formation of carbon
mass.
dioxide?
Oh, for carbon dioxide, you can add a gas syringe on
That,
top.
that's correct. We can use a gas syringe and we can note down the change in volume per unit time for the amount of carbon dioxide that is produced.
Yeah.